So let's continue with the cell cycle. And in the previous class, we had talked about the preparatory phase before the actual cell division happens. And we said that the cell cycle has got the M phase, that's actual cell division, which is either meiosis or mitosis, and the I phase, which is the interphase. So we talked about the three phases of interphase, G1, S, and G2. And of course, there are some cells which would prefer to be resting, not dividing, like the nerve cells. That's the G0 phase. And then after we have prepared, we have divided the DNA. Now we are ready to form another cell. That's when we are going to step into meiosis and mitosis. So let's start first with mitosis. So what is the difference between mitosis and meiosis? Well, the difference is easy. When you're talking about mitosis, this cell division is going to happen for all other cells except gametes. Meiosis will happen for gametes. Easy, right? So let's go. What happens is that the first phase of the cell cycle, of course, we know is interphase. That is preparatory. After you have prepared, you have divided you've made sure that the organelles have been replicated, you have formed proteins, you have replicated the DNA, then you can go into the actual M phase, which is the actual cell division. So mitosis is a process where a single cell divides into two identical daughter cells. Please take note, right? Single cell or single, right? Single cell divides into identical. Take note of the identical there. Okay? Daughter cells. Identical to daughter cells. So the thing there is there are two. Okay. So two identical daughter cells. So imagine you have got a cell. They told you it underwent four mitotic divisions. How many cells are you going to have at the end of the day? So this is a cell. After the first division, you have got two. Okay? You have got two cells. After the second division, you are going to have four because each one of them is giving you two. After the next division, you are going to have what? This is going to give you two, two, four. So you are going to have eight. Okay, like that. So you see how many divisions are here. First division, second division, third division. So what do you think is the equation depending on the number of divisions to find out how many cells? So the number of cells that you have is going to be equal to 2 raised to the power n. If you have got no division, you only have one cell to the power 0. If you have got two divisions to the power 2, the answer is 4. Two, if you have got, sorry, one division, the answer is going to be 2 cells. If you have got two divisions, you are going to have 4 cells. If you have got 3 divisions, just like that. Please take note of that. There are some nice questions that you can just find. They pop up in the test or the exam. All right. So when it comes to the phases, I remember the phases of the cell cycle myself as IPMAT. IPMAT. The first phase is interphase that we have already discussed, which is the preparatory phase. The next will be the prophase, metaphase, anaphase, and then telophase. IPMAT. Okay, so starting with the first phase, in this case, that is the prophase. What is happening then in this phase? So not anything complex. What is happening basically is that in prophase, remember you have got, this is your cell which has undergone all the cell division, the nucleus, the DNA in the nucleus has also divided. But then the thing is that All right, so the thing is that we don't know if we have got the DNA replicated. And I'm saying that because you cannot see the DNA really when you look at the microscope. Okay, you can't see it. So in prophase, what is going to be happening is that that DNA that you can't see, you start seeing it. Okay, you are going to start seeing it. So the nucleolus, this thing that is surrounding the nucleus, is going to disappear. No, first the nucleolus is not what surrounds the nucleus. When you have got a nucleus, the middle of the nucleus, which is dark, is known as the nucleolus. So that is going to disappear. And then after then, the next thing that we are going to be able to see, so the nucleolus disappears. The next thing is that the replicated chromatin is going to condense. Okay, remember it replicated in the previous phase, which is the interphase. So replicated chromatin condenses. Initially, when you look at a microscope, you can't see the chromosome, but then when cell division starts, because of the condensation of the chromatin, you can now see the chromosomes on the microscope. So replicated chromatin condenses. And then after then, the next thing is going to be, of course, this chromatin that has replicated, take note. Remember the chromatin has got what? It is replicated. Thank you. It has got sister chromatids, right? Yes. And then the third thing is that at the centromere region of each chromosome, a large protein complex called the kinetochore is a site of attachment to the macrotubules. So I've got a kinetochore here. That's where the microtubules are going to be able to attach later on. The centrosomes with their duplicated centrioles separate and migrate to opposite poles of the cell. Take note, that's the centrioles. Okay. First, you have got a centrosome. So centrosome with their duplicated centrio are now going to separate and migrate opposite poles. You understand what these centrioles are. Okay, so that is what is happening there. The microtubules or the mitotic spindle will polymerize between the two centrioles. So you are now going to see the microtubules, and then you are going to see some other things that will be forming. So basically, what are we saying? So the nucleolus has disappeared. Okay. Also, the nuclear membrane will also disappear. The nucleolus and the nuclear membrane will disappear, and then the spindle fibers move close to each other or close to the chromosome. Spindle fibers move close to the chromosome. Okay, that's what is happening. And then the centromere of the chromosome develop an attachment point that I've already mentioned. Let me write it in another color just to emphasize. So the kinetochore is the attachment point on the centromere. kinetochore. There, that's the attachment point on the centromere. So these are the changes that you'll be able to see in the first phase, and that's in the prophase. Okay, nucleolus disappears. So if this is your cell, you cannot see the nucleolus, neither can you see the nuclear membrane. The chromatin condenses. Now you can see like the chromosomes. You can see the chromosomes which have duplicated. You can see them. You see them. They are of course not arranged centrally, but you see them like that. Centrosome with their duplicated centriole separate and migrate to opposite sides. So we're going to see centrioles on opposite sides. We'll call those as centrioles on opposite sides like that. Spindle fibers move close to the chromosome. Spindle fibers, these are the things that will be able to attach to the chromosomes, but this is the first phase. Okay. And then the second phase, remember IP, mat. So the second phase is the metaphase. So how do you remember meta? M for mid. So M for middle. What is going to happen is that the chromosomes are now going to line up in the equatorial plane. Remember, they are aligning. In the equatorial plane. Okay. And their double structure is now visible. They are now visible. You can now see them. The next thing that happens is that each is attached by microtubules extended from the centromere to the centriole, forming the mitotic spindle. Okay, they are attached. For that one, I'll not even write that point. And then chromosomes will further condense and attach the mitotic spindle at large kinetochores. So that's the next thing that you'll be able to see. So we're going to see this is a cell. And what you have are chromosomes on the middle here. Remember on the sides, what do you have on the sides? Excellent. So you have got these guys, the centrioles. So they are going to send spindle fibers. These spindle fibers are attaching where? On the kinetochore, which is on the centromere, right? So the mitotic spindles are going to attach like that. Going to attach like that. Okay? So there, that is what we form at this point. So microtubules extend from the centromere to the centriole. So this is a centriole. And these are the spindles, spindle fibers or mitotic spindles. Okay, and then they are going to attach to the kinetochore. So you can see they are aligned on the equator because this is the middle, that's known as the equator or the equatorial plane. After the metaphase, IP mat, remember the next phase is the anaphase. Now in the anaphase, just think of A for away. So now these guys are going to be separated. Okay, the spindle fibers will be able to pull the identical chromosomes apart, breaking the centromere. So spindle fibers pull. Okay, these are going to pull identical chromosomes apart. and then you are going to break the centromere. So what do you think is now going to happen? So you have got still this as your cell, and you are having the mitotic spindles going like that. And then what has happened is that you have put these guys and they are away from each other. You have put them. You can see how they've been separated. So that's anaphase. Okay, basically that. And then the next phase, which is the last phase here, is telophase. And can you guess what is going to happen in telophase? So in telophase, think for T for 2. So in telophase, each end of the cell now contains identical chromosomes. So the things that had disappeared in prophase will now start to appear. So nuclear membrane and nucleolus reform. Okay, nuclear membrane and nucleolus reforms. Apart from that, what else is happening? So these guys will now surround. These guys which have reappeared will now surround each, okay, chromatid. Remember, chromatid is after the chromosomes have separated, right? Yes, so then each now is going to be surrounded. So basically that is what is happening, and the chromosomes will uncoil and again decondense. Okay? Remember, they are decoiled in prophase. Chromosomes uncoil. Uncoil and decondense. It means that if you look on a microscope, you can't see them anymore again. Yeah. And that is because they are preparing for another interphase. So what you are going to see in this phase is you have got your chromatids there. And then these guys have been surrounded by the now appearing nuclear membrane. You can see we have separated the nuclear. You have got now two nuclei. This is known as karyokinesis because we have now separated the nuclei, karyokinesis. But then we need to form two different cells, and that is going to take us into what? Cytokinesis. Okay, what will happen is that we'll start forming a few row there. Depending with what type of cell you are talking about, if it's an animal cell or if it's a plant cell. So there's going to be a constriction that is going to be happening. And that constriction is what is going to end up forming two different cells. And that one we are calling it as what? That's known as cytokinesis. So we are going to form a cleavage furrow. Please take note, depending with what cell, the name is going to be different. Okay. So cytokinesis is going to be the last phase. So cytokinesis in plant cells has got a name, and in animal cells it has got a name. So in animals we are calling it a cleavage furrow. Okay. And then in plants, we are going to call it a platter. Okay. So basically, that is what is happening in these phases. Can we go through a real quick one? So the first thing that is happening, what is happening first? We have got replication of the cell. After that, we go to the interphase. What is happening in interphase? In interphase, there is just a disappearance, okay? The disappearance of the nuclear membrane, the disappearance of the nucleolus, the condensation of the chromosome so that it can be visible. We go after prophase, we are going to go to metaphase, the chromosomes realign on the middle, anaphase, they are pulled away, telophase, they are separated, you have got two. So that is mitosis. So the next phase we should understand is meiosis. Now take note that mitosis happens only in somatic cells. Remember, what are somatic cells? These are basically body cells, not every cell, only the body cells, because the gametes don't undergo mitosis. What they undergo is meiosis. So let's talk about meiosis. Now, meiosis is where just similar to mitosis, with a difference of meiosis has got two phases. There is meiosis 1 and there is meiosis 2. That's the difference. Okay, so when meiosis 1 is done, then we are going to have meiosis 2, but the phases are the same. We also use IPMAT, but you'll be putting 1 and 2. Interphase is the same for meiosis 1 and also for meiosis 2. So let's just go on with prophase. Okay, but take note that meiosis in which cells? It is only in gametes, and these gametes undergo sexual reproduction, because mitosis is asexual. You have got one cell, it is dividing without there is no woman, there is no man. But for meiosis, you're supposed to have a man and a woman. So that's a mode for those cells that undergo sexual reproduction. After cell division, I've got 23 chromosomes at the beginning, because for mitosis, if you started with 46 chromosomes, after cell division, you still have 46 chromosomes. But in meiosis, this is what important. When you start with 2n, which is haploid, you are going to end up with, this is diploid, you are going to end up with haploid, which is just n. So this N now for a man should combine with N from a woman to form back the 2N for the child. So that's why it only happens in gametes. And take note that unlike mitosis, where the offspring identity, here there is genetic variability. Okay, that's important. Genetic variability. It's actually meiosis that explains why you don't look like your siblings, because of the cell division that was happening. So the cell division that was happening made sure that each sperm contains different genetic information, and so when crossing over gets to happen, I mean when intercourse gets to happen, you have got a different genetic makeup. Okay, so that is what happens. And this is going to happen between gametes, as we've said. In male, the gamete is the spermatocyte. In females, it's the oocyte. Before meiosis occurs, you're supposed to have interphase to prepare, and you know the phase of interphase, you have got the G1S and the G2 phase. And then now you're going to have the different phases now of meiosis. Okay. So what is going to happen? Let's go to the first phase, which is prophase 1. So prophase 1 should be the most important phase when you are talking about meiosis, because there are a lot of new things that are different from the other phases. Okay, it's prophase 1, different from the other phases. But then why do we even let a cell to undergo cell division? So there are four main reasons: for growth, for repair. Let's say cell died, you need to repair it. Also for replication, okay, or depletion of the loss. Like if you lost a cell, it needs to be replaced, and also for reproduction. Now, when we go to meiosis, starting with prophase 1. Prophase 1 is special in that, let's first talk about what is going to happen. All the same phases that happened during prophase of mitosis is what to happen here. There is the disappearance, the condensation of the disappearance of the nuclear membrane, the disappearance of the nucleolus, the condensation of the DNA, and those things. But except the prophase 1 is divided into five phases. Okay, it's divided again into five phases. So I've got what we call leptotene, zygotene, pachytene, diplotene, and diakinesis. Those are the phases that we're going to find in prophase 1. But then I'm going to waste no much time on those phases, okay, because at your level there is not much. So let's go to prophase 1. So in prophase 1, what we are seeing is that similar to what was happening in the previous one. So what was happening, I'll not even write it because the chromatin is going to condense into chromosome, the nuclear membrane along with the nucleolus is going to disappear. But the major difference that happens is that there is genetic recombination. The reason as to why you are different from your siblings happens here: genetic recombination. This is the most important. Okay? Genetic recombination. The very, very most important. So, what is happening in this process of genetic recombination? So, genetic recombination is happening by a process that is known as the crossing over. This happens by what? By crossing over. But really, what is happening? Just to remind you, remember before prophase, what is the phase before prophase? IPMAT. It is interphase. So in interphase, what happened is that we add this one cell. Okay, we add this one cell. Let me write another one. We also had this cell. But then what happened is that these two cells in interphase made a copy of themselves. And when they made a copy, that was during replication. Let me write this guy here. Okay. So this phase is supposed to be what? Replication. And replication is not part of prophase. This is actually interphase. This is the I phase. So after replication, what you see is that this guy made a copy of itself, and then the other guy also made a copy of itself. But then what is going to happen now is that in prophase 1, now prophase is going to start. Okay. So in prophase, the first thing that is going to happen is the forming of a synapse. So these two guys that you saw like this, I'm going to be consistent with the color that I'm using for both of them. There's going to be what we are calling as a synapse. What is a synapse? Synapse is just coming together of the homologous chromosome, because these guys are actually homologous chromosomes. Okay. They are homologous chromosomes. Initially, they were together like this. Let me even actually even put them together. They were like this. This guy here and this guy here. These are homologous chromosomes. So even when each one of them made a copy, they didn't stop being homologous. They are still homologous chromosomes. So when they come together in synapse, they are going to stand next to each other as homologous as they are. So you have got four homologous chromosomes. They are still homologous. It's just that each one of them has got a copy or a sister. Okay. And then after this phase, the next thing that is going to happen is something interesting. And that is what causes what we call genetic recombination. So look at what happens here. Can you see that? This guy has undergone what we call crossing over. Okay. At this point where there is crossing over, that is known as a chiasma. That point where they have connected, that's known as a chiasma. Okay, that's a chiasma. And we are calling this phase as a synapse. This is also synapse because they are still close together. Okay, with a chiasma at this point. Now, these guys, when they form a chiasma, you can see they are four and they are together. We call this as a tetrad. Tetrad. A tetrad has got four chromatids, but it has got two chromosomes. So as well call it as a bivalent. Bivalent refers to the chromosomes. I only two. Tetrad means the sisters, the chromatids. They are four. But take note that the blue ones are sisters to each other, and the other color they are also sisters to each other. They are not all sisters. Two are sisters, two are sisters, but the two, this one big one and this one big one, they are, if you put them together, they are homologous. So after this synapse, what is going to happen is that the genetic recombination, there's going to be the sharing of information. So this blue one is going to be like this. And then the other guy is going to be like this. What has seen has happened. So that is known as this phase that has happened here. That's known as crossing over. Because some information which was on one chromosome has gone on to the other, and what was on the other chromosome has gone on to the other. That's crossing over. And it is for this reason that after cell division, because these two guys are supposed to split like this one will go into one cell, the other one will go into another cell. So because they are now different from what we started with, that's why the offsprings will be different after intercourse because of the crossing over that has happened. So after crossing over happens, we produce what we call recombinant chromosomes. So these guys that you can see here, these are known as recombinant chromosomes. Why recombinant chromosomes? Because they are formed after genetic recombination. Okay, and these guys are unique, and the sisters are no longer identical. So you now have an identical sisters. Initially, the sisters were identical. You can see these two are sisters, they're identical, but then the sisters are no longer identical. So basically, this is what is happening during prophase 1. And the rest of the steps are going to be the same, all right? From metaphase up to the last step, they'll all be the same. So, after prophase, the next one is what? So let me now put them. So This is the one. I'm just going to write. Of course, we have got something like this. Okay, you can see after genetic recombination. And after the next phase that was supposed to happen was what? After prophase, it's metaphase. Okay, so it's metaphase. Now, these two guys were supposed to be sisters, isn't it? So they're supposed to be sisters. No, not really sisters, but they're supposed to be together. They are homologous chromosomes. So this is how they're supposed to be in a cell because they're homologous. So what is going to happen is that you have now attached these using spindle fibers. They are aligned on the center. So this is metaphase and it is metaphase 1. So in metaphase 1, what can you see? It is homologous chromosomes which are on the center. After metaphase, what do you have? It's anaphase. So in anaphase, we are now going to be separated to two ends. Okay, so you can see there, you can see they've been put to two ends. So what I want you to understand is this. In anaphase 1, what is separated are homologous chromosomes. Okay? Homologous chromosomes. So homologous chromosomes have been separated in anaphase 1. That's what I want you to know. And then the next phase now after anaphase 1 is going to be, we will step into telophase 1. So telophase now will make two cells. What you see is that in each cell you're going to have a blue, which has got, of course, another gamma part there. So we're going to see in each cell like this. So this is telophase 1. Okay, so basically this is what happens. And the most important thing that you need to know is that it is actually in telophase 1, this is very important, that you change. The cell that you started with was initially 2n, but it is now only n. So in other words, you are changing a diploid cell into haploid in telophase 1. And then the rest of the things will be the same. Nucleolus is going to reappear, the the nuclear membrane is also going to appear. There's going to be decondensation of the nucleus of the chromosome. I mean, everything else will go back to normal. Okay, and then we'll step into now the second meiotic division. So when we go to M2, second meiotic division, we're now going to have prophase 2, prophase 3, and prophase 4. But then for the 2s, there will be no more like the previous one. You can see now this cell has got only one chromosome, right? Even this one has got one. So what is going to happen is that here, so you have got now a chromosome there. You can even have another chromosome there. Don't get confused. I could have also put other chromosomes like that, which we could have separated. Okay. Those were homologous chromosomes. But here in prophase 2, actually prophase, the same thing will happen. In prophase 2, there's going to be the disappearance of the nuclear membrane, there's going to be the disappearance of the nucleolus, and then the condensation of the nuclear, okay, the condensation of the chromosome. And then that's when now we're going to have metaphase 2. Guess what is going to happen in metaphase 2? You have got sister chromatids. And this is important. In metaphase 1, what was on the middle, what aligned on the equator was homologous chromosomes. Homologous. But these are sister chromatids. Okay? And then the next phase is going to be anaphase. Anaphase 2. Please take note that while in anaphase 1 we just separated homologous chromosomes together, it is in anaphase 2 that we are breaking the centromere. You can see the guys which we are held are the centromere. We are now splitting them. We are now splitting them. So this is a totally different thing. Please take note. What are we splitting? We are splitting sister chromatids, not homologous chromosomes, sister chromatids. And take note, sister chromatids are held by a centromere. So we have broken a centromere. So it is actually prophase 2, which is so similar. Sorry, metaphase 2, which is so similar to mitosis that we talked about. Okay. So, and then in telophase, the same thing that happened during meiosis is mitosis is going to happen. You saw that, yeah, the same thing that happened in mitosis is going to happen. We are now going to have two cells, and each one of those cells has got half the number of chromosomes, 23, 23. Okay. But take note that the change of chromosomes happened in telophase 1 from 2n to n. So basically that is it when it comes to the cell cycle. Let's answer some questions and then we are done with the cell cycle. Explain what happens during G2 phase. Chromosomes replicate. Is that true? No, there's no replication of chromosomes. DNA replicates. No, that's S phase. Enzymes and proteins are synthesized. This is true, right? It's proteins and organelles which are made. A cell experiences growth. Of course, that's also true. A cell is experiencing growth, isn't it? And then the next option, 2 and 4 above. So meaning 2 is DNA duplicates. Now DNA replication doesn't happen in G2. DNA replication happens in which phase? That's in the S phase. So in G2, there is the synthesis of protein, organelles, and components needed for cell division, just like the G1, right? So yeah, that's what happens. So enzymes and a cell experiences growth. Of course, that's why it's called the growth phase. Yeah. But I wonder why there's no answer for both 3 and 4. Enzymes and proteins are synthesized, that is okay. A cell experiences growth. Of course. But the best that since there's no answer for both of them, we can go for enzymes and proteins are synthesized, because it is in G2 that we have synthesis of proteins, organelles, and other components needed for cell division, just like in the G1 phase. But the G1 was mainly for growth. And in G2, we want to see that anything that was not made is now made, or anything that was left out is now made. Okay. So hope that is clear. So then what is the answer here? What is the best one we can go for? It is enzymes and proteins are synthesized. In mitosis, chromatids are held together by what holds chromatids? These guys, like, so what holds them here? It's a centromere, isn't it? These spindle fibers, the centrosomes, the centrioles, and the microtubules, these are just going to be involved in getting to pull the cell away to pull the chromosomes away during cell division. Choose the event which corresponds to prophase. Formation of a wall plate, is that true? No, wall plate, this is cytokinesis in plants, isn't it? So no. Formation of four haploid cells, is that true? Haploid cells, are we forming them in prophase? No. Formation of spindle fibers, are we forming them in prophase? Okay, let me put a dot. Alignment of chromosome along the equator, that's metaphase, right? Separation of sister chromatids. Do we separate them in prophase? No, we don't separate them in prophase. That's in anaphase. Okay. So it's formation of spindle fibers. Remember we said that the spindle fibers are going to form and then they'll come and attach on the kinetochores, and then that's when there'll be alignment on this end. Cell plate formation is a feature of bacterial cell division? No. Plant and animal? No. Animal, we said animal is cleavage furrow, it's only plant. Okay. Separation of bivalent takes place during, bivalent? Yes. A bivalent is these guys, these two. When do we separate these two guys? In which phase? Homologous chromosomes. It's in, remember anaphase is for away. Anaphase 2, not 2. Anaphase 1. Because in anaphase 2, what we are separating are the sister chromatids. Cytoplasmic division of a cell is called, thank you, cytokinesis. Choose a stage when the diploid cell becomes haploid. Thank you, telophase 1. Choose a correct statement. The cells from which gametes are produced are haploid. Is that true? Okay, I'm gonna put a dot there. Sexual reproduction provides for genetic variation. Is that true? No, sexual reproduction, there's no genetic variation unless asexual. Remember, oh, this is actually correct. The asexual is for mitosis, and that one doesn't provide for genetic variation. But sexual is meiosis, and we have already explained that meiosis provide for sexual, for genetic variation. That's why you look different from your brother, right? So I'm gonna take this one. The male reproductive organs are called gametes. Is that true? You would say true, but no. These are not reproductive organs. These are gametes, are not organs. These are cells. Okay, like the sperms and the ovum. They are not organs. They are reproductive cells. Mitosis produces a tetrad. No, tetrad is involved in meiosis. Meiosis is important because it is responsible for repair of broken tissue. No, it is about giving birth. Repair is mitosis. Remember, mitosis is just cell division, right? What you have, you want to divide so that you can have another one, right? So the cells which gametes are produced, the cells from which gametes are produced are haploid. No, gametes are produced, if you saw in meiosis, gametes are produced during meiosis. And I mentioned to say meiosis, we first start with 2n. The 2n is now going to convert to 1n. That 1n that we are forming later on, that means that we started from diploid, and then in telophase 1, that's when we ended up having haploid. Choose a false statement. Synapsis is a regular occurrence in meiosis. I'll put a dot there. Mitosis is a reduction division. This is true. It's reduction. Like if you start with 2n, you are going to go to n. That's reduction. Male reproductive organ produce gamete. Okay. Male reproductive organs produce gametes. This is true, right? Like the testes who produce gametes, which are the sperms. The genetic content of tissue cells are identical. Genetic content of tissue cells are identical. Yes, tissue cells, these are somatic cells. Their genetic makeup is identical. Like somatic cells, they've got the same genetic makeup. So if your leg, the cells in your leg, the chromosomes there is saying that on this position you're supposed to have a blue eye color. Any other cell is going to have the same, that has got DNA is also going to have that same information. So number one, synapses is a regular occurrence in meiosis. Yes, synapses should always occur in meiosis. So all of the above statements are false. Choose the first statement. Not that all of the above statements are false. That's not true. Okay, let's go. So all of the above statements are false. Not really. This is not true. Okay. That would even be the answer. For all of the above statements are false, because not all of these are false. Okay. Now let me answer this question properly, because the way I'm answering it is, so number one, synapses. We want a false statement. Synapses is a regular occurrence in meiosis. This is supposed to be regular, supposed to be always happening. Mitosis is a reductive. This is true, it's not false. Male reproductive organs produce gametes. That's true. The male reproductive organs produce gametes. But I'll put a dot also there. Genetic content of tissue cells are identical. That is true. Now, the male reproductive organs, even the statement, the male reproductive organs, this is too generalized. But what will produce, of course, the gametes, because the male reproductive organs are a lot. That is ranging from the penis you are having, of course, the urethra inside, and this is going into the spermatic cord. You have got, of course, the scrotum here, you have got the testis, you have got the seminiferous tubule, and then you're going to have the spermatic, I mean the epididymis. So not all of these will produce gametes. Gametes are specifically produced here in the testis, specifically by the seminiferous tubules. So if they wanted to be specific and they are saying organs, then I would remove this one. Okay, hope you get it. Otherwise, the other options are making sense for me. Chromosomes are best observed at the stage of, which stage do you observe chromosomes? Remember we said that the chromosomes will start condensing in prophase, but when really do you see the chromosomes? So even when the condensation is starting in prophase, of course, yes, in prophase they are going to condense, and but they are going to line up on the middle by the metaphase anyway. So prophase is when they condense, that's when you can see them. In anaphase 1, the chromatids are pulled apart. That's not true. Anaphase 1, you pull apart what? Homologous chromosomes. Centromeres are replicated. No. In anaphase 1, remember, anaphase is pulling apart, not replicating. Chromosomes exchange segments of DNA. This is true. That's where crossing over happens in anaphase 1. Non-sister chromatids are separated. You don't separate the non-sister chromatids, you separate the homologous chromosomes. Centrioles replicate, no. Somatic cell has 24 chromosomes aligned at metaphase. How many chromosomes are present at anaphase? Remember, this is a somatic cell, it's a body cell. So what phase do you think is happening here? This is mitosis. Okay. So if in mitosis, at metaphase you have got 24 chromosomes, how many chromosomes are present at anaphase after separating them? So think of this. You have got this. These are three chromosomes. If you separate them, they are going to be, you're going to have this. So how many do you think there are now? So there are still 24 chromosomes, because by this point they are not yet, these are sisters, they are chromatids. They have not yet condensed, but of course, if we can just to be try to be a bit more specific, this is going to be double the number, because when you separate on the middle here, this will be a different cell, this will be a different cell, so they are going to double. So 24 plus 24, that's 48. Although you don't yet have two different cells, but because of the replication. Interphase is the part of the cell cycle when a cell ceases to function. No. A germ cell forms a spindle apparatus. Not really. That's anaphase, right? That's prophase. A cell grows and duplicates its DNA. That's true. Mitosis proceeds. No, it precedes, because mitosis is behind. It's the first. Actually, interphase is the first, and then mitosis. Meiosis takes place. No, meiosis doesn't take place here. So a cell will grow and duplicate its DNA in interphase. Crossing over occurs during. In which phase does crossing over happen? Lactotin, Pachitin. So this was what I was trying to mention. Unfortunately, it has come. So let me just mention them. So when it comes to prophase 1, prophase 1 has got 5 more phases. Okay. So the first one is known as leptotin, in their order: leptotin, that's number 1. The second one is zygotin. The third one is pachitin. The fourth one is diplotin. And then the last one is diakinesis. If you want to remember, you can just say diakinesis. Let Zambia peacefully dance daily. Can you remember that? Huh? Let diakinesis. Let Zambia peacefully dance daily. But you have got diplotin and diakinesis. So how would you know dance daily? Which D comes first before the other D? So just think of all of them are ending in tin except the diakinesis. So leptotin, zygotin, pachitin, diplotin. The last one is anyway something else, diakinesis. So in leptotin, the main thing that is happening there, leptotin, what is happening is that the chromosomes will start to condense. Okay, chromosomes condense, and then you are going to be able to see them under a microscope. And the nuclear membrane will start to break down. In zygotin, remember in prophase, this is prophase 1. These are the phases of prophase 1. And that's where crossing over happens. So it's in zygotin that homologous pairs align, pair up and align, forming structure called the synaptomere complex. Okay. So it is in this phase that these homologous guys were going to align. And then in Pachitine, that's where crossing over occurs. So here, that's where the crossing over is going to occur, like that. And this process is going to be the one that leads to the genetic recombination with a chiasmata being formed there. Diplotin homologous chromosomes start to separate but remain attached at the chiasma. Okay. Remember, it's homologous chromosomes. They'll start to separate, but they are still attached here. So they are coming away from each other. They are moving away from each other, but then they are still attached on the chiasma. And then dark kinesis, the chromosomes are fully condensed and the nuclear envelope completely break. Chiasmata move towards the end of the chromosome. Okay, so this is now the final. There is complete everything. So crossing over happens during which one? It's the crossing over is happening during Pachitin. All right. Linkage prevents homozygous condition, heterozygous condition, recombination, segregation of alleles, coupling, and repulsion theory. So linkage, homozygous, heterozygous, it doesn't prevent recombination. That's actually what's happening. Segregation of alleles is what is happening. We're not preventing it. Hetero, so we actually prevent homozygous condition where everything is going to be the same, because without genetic recombination you would all look the same. Chromosomes that are of the same size, shape, but pattern are called homologous chromosome. Process of matching chromosomes becoming zipped together occurs during, remember, matching chromosomes becoming zipped together. It's where prophase 1, this is not A, or that's 1. During which of the following stages of cell cycle would the chromosome consist of only a single chromatid? Remember, only a single chromatid. Okay. Karyokinesis, the separation of the nucleus, mitotic phase, metaphase. In metaphase, no. Prophase, no. Because you have got both chromosomes. You have got the sisters, I mean. G1, not really. G2, not really. It's in karyokinesis. Karyokinesis when you're separating the... Okay, let's see again. Karyokinesis is when you're separating the nucleus. Yeah, when you're separating the nucleus, each chromosome has got only one cell, because remember you add this, right? They have replicated. This is your cell. After you separate the nucleus, then each one of these is going to go in its own nucleus. So that is happening in karyokinesis. Which of the following occurs in meiosis but not in mitosis? We want in meiosis segregation of sister chromatids. Okay, let me put a dot. Pairing of homologous chromosomes. It happens everywhere. Crossing over between homologous chromosomes. This one happens only in meiosis. Alignment of chromosomes on the metaphase. This one happens for both 2 and 3. Pairing of homologous chromosomes happens for both. So this one can't work out. Okay. So remember, if it is in meiosis, you are going to have, now what you pair, actually pairing of homologous chromosomes, what you are pairing in prophase of mitosis, you just have sisters. They are not homologous chromosomes, they are sisters. Yeah, so there's no pairing of homologous chromosomes. In the life cycle of a human cell, each chromosome contains 2 chromatids by the end of the dash. Life cycle of a human cell, each chromosome contains 2 chromatids by the end of G1, not really. G2, not really. Metaphase, not really. Cytokinesis, not really. Synthesis, that should be synthesis, right? DNA synthesis. But they've just said synthesis. What synthesis? Because each chromosome will contain 2 chromatids at the end of, like this is a chromosome, to undergo replication. And this replication is happening in the synthesis phase. That's the S phase. Remember the S phase. Then you are going to have this. Study the diagram below which shows some stages of mitosis. Identify the stages labeled X, Y, and Z. What do you think this is X? And then Y, and then Z. So X, these things are just confused like that. Okay, that can be interphase. And then Y, you can see they are on the sides, and it looks like two cells. Actually, what looks like interphase would be this one, because the cells are just confused like that. I mean, the chromosomes are just all alike like that. So that looks like interphase, but then is apart from interphase, you can as well have prophase, which is like that. Prophase, that's when you start seeing the chromosomes. So where is prophase? Prophase is here. But can this be interphase? That disqualifies this option. It can't be interphase. So we go to the next one that looks close. What does Y look like? It looks like the two guys are on the two ends, isn't it? That's like anaphase, but then it can't be anaphase. It looks like this is on the middle. Actually, they've been separated. This will go into one cell, this is another cell. That looks like telophase because you can see two on two ends. So if it's telophase, it can either be this one or that one or this one. Okay, so meaning again, we are going to disqualify this one already. It can't be an answer. And then we remain now with the three. So if you look at Z, all these options they have got, all the options I've ticked, they have got metaphase, anaphase, metaphase. So can this be metaphase? This is on the middle. Can this be anaphase? This can't be anaphase, so we'll remove this. Because anaphase, they're supposed to be away from each other. So it should be metaphase. And then what would be X? If these two are metaphase, these two are appearing. Is this anaphase? This looks like anaphase. Can you see that they are away from each other? Anaphase. So best would pick is that one. Okay. Synapse is the process of during mitosis. Just mitosis makes this answer wrong. During meiosis, whereby sister chromatids exchange their material. It's not sister, it's the homologous. Okay. These two guys which are crossing are not sisters. The sisters are these two and these two. Whereby daughter cells with half the genetic material of the parent cells are produced. That's not true. Whereby homologous chromosomes separate and migrate to opposite poles. That's not true. It's not separating. Homologous chromosomes becomes closely associated. That's true. That's synapsis. And the last question. Study the figure below and answer the questions. So can you see what's happening? The letter U in the diagram 2 represents, you can see this is the diagram 2. This is U. It's the chiasma there, right? A chiasma between 1 and 2. Okay. 1 is this one. 2 is this one. So it's not a chiasma between 1 and 2, but 1 and 3. Can you see that? It's between this guy, this one, and also this one is 1 and 3. So that's the answer, this one. The structure in diagram 3, this was supposed to be 3, represents, what are we saying? This has already undergone genetic recombination, a chiasma. It's not a chiasma. The behavior of chromosome during metaphase, no. It's not a chiasma. Behavior of chromosomes during anaphase is genetic recombinant gametes. Yes, they have undergone recombination. If you can see, there's a combination of these chromosomes. So that's recombinant gametes. So that is it actually for this class. Thank you so much for having been present. Stay blessed.